Hey CW Bio, Mr. Kennedy here. Today we're going to talk about mutations, which is part of your Chapter 12 DNA lecture. We've been spending a past couple of videos talking about DNA structure, what it looks like, how it's assembled, and finished by going through and looking at how DNA copies itself. So today we're going to look at this idea of mutation, accidental change to our DNA that can occur anytime DNA is manipulated in any way, shape, or form. So through DNA replication, sometimes mistakes get made. From cell division, as genetic material is passed from one cell to the next, sometimes mistakes are made. Doesn't matter if we're talking about a little mistake or a big mistake involving maybe just one nucleotide or a whole chromosome. It's all mutation, and it all can have an impact on what we see outwardly, both in the mirror and even through our biochemistry, creating genetic disorders. So when you think about mutation, the whole idea of mutation is that we're changing our DNA sequence. So as you can see from this slide, any change in a DNA sequence is called a mutation. And because your DNA is so special, your cells, your body has actually evolved multiple ways to protect your DNA from these changes. After all, we want you to, reach your full potential and to work at maximum efficiency. So in order to do that, we need to protect your DNA. And at the bottom of this slide, you can see that mutations can be caused by errors in replication, transcription, cell division, or even external agents that we get exposed to. Let's explore this in a little bit more depth. So mutations, they can affect both body cells and reproductive cells. If it affects a body cell, then that mutation might lead to problems, just for you. But if it affects a reproductive cell, that means it could be passed on from one generation to the next and affect your offspring. Mutations can affect reproductive cells by changing the nucleotides within a gene in a sperm or egg. And if that sex cell, gamete, takes part in fertilization, then that gene would become part of the genetic makeup of our offspring, and that's how these mutations get passed on. If it's a mutation that happens to me, like let's say I go out and stand in the sun too long and end up with a mutation in my skin cells that creates skin cancer, that's my problem. I'm the only one that's going to have to deal with that, right? But if it's in the reproductive cells, then offspring, future generations are going to have to deal with that. Sometimes mutations, instead of just causing negative things like cancer, can actually produce things that are, theoretically good. They could produce a new trait or create a new protein that didn't exist in the parents. Now, statistically speaking, the odds of a random mutation creating a new characteristic that is actually advantageous to your survival are pretty small, but the possibility is there, okay? The flip side is the protein that doesn't work correctly, and that's where we got to talk about things like cancer, an embryo that doesn't survive, all of those sorts of things, okay? So there are both good mutations and bad mutations, depending on which one of these you're looking at. Now, last thing we got to kind of deal with is where do these mutations come from, and what do they look like? So the first thing that I'm going to tell you is that anything that causes a mutation, it kind of falls under this umbrella term, mutagen, okay? So if you create a mutation, you're a mutagen. So here's an example. What happens if powerful radiation, such as gamma radiation, hits the DNA of a non-reproductive cell like skin, muscle, or bone? At the end of the day, that's going to create a mutation in the DNA in your skin, muscle, or bone. That gamma radiation is considered a mutagen. Now, what kind of mutation you get, whether it's good or bad, leads to cancer, or nothing happens, will determine whether or not we change the mutagen word to something like carcinogen. If that mutation leads to cancer, then we can call it a carcinogen. If all it does is create a mutation, then we just stick with the word mutagen, and that's the end of the story. keep in mind that if this mutation again happens in a non-reproductive cell, well, then I'm the only one that's going to have to deal with it. The mutation, as it says at the bottom of this slide, may cause problems for the individual, but not for future generations. Okay? If it happened in the reproductive cells, again, that's where future generations are going to have to deal with it. Now, this damage, well, it's something that your body's going to try really, really hard to protect you from. So damage to genes, they can impair the function of the cell. When the cell divides, cells, or that are produced by that cell are also going to have the same mutation. And so that mutation can literally spread throughout your body cells. So there's proofreading and corrective processes that go on, like gene, like editing sequences that will go in and cut out mistakes and replace the damage that's been done to help protect you from this stuff. And it works really, really well, especially when you're young. Especially when your exposure to a mutagen is small or maybe only for a short time period. The older you get, the more you've been exposed to these mutagens, the harder it is for this editing and repair process to keep up and to prevent the damage from spreading, right? So if we damage a gene in a given cell from exposure to gamma radiation, that one cell divides. Now we've got 2 cells that have that damage. Those 2 cells divide. Now we have 4, so on and so on and so on, and it can multiply out. Some mutations of DNA in body cells can actually affect the genes that control cell division. And if that happens, that's where the cancer is involved and our mutation becomes carcinogenic. Okay. So one cell divides into 2 cells to 4 cells to 8 cells to 16 cells in a very, very rapid fashion because the cell division cycle is basically running out of control. So that's how we define cancer, right? Uncontrolled cell division, which could cause from exposure to a mutagen that damages the protein that controls the cell cycle. Let's look at some specific types of mutations and their outcome. Okay, we're going to start here with what's called a point mutation. Point mutation is a change in a single base pair in DNA. So basically something like gamma radiation or any other type of mutagenic agent comes in and it's going to simply wipe out an A, T, G, or C that we would normally expect to be in one locus or location in your DNA strand, and we're going to end up getting something else, right? A change in a single nitrogenous base can actually change the entire structure of a protein, but the reality of it is it really depends on where that point mutation happens. As you'll learn as we get deeper into transcription and translation, or basically protein synthesis, each amino acid in a protein sequence is coded for by 3 separate bases in DNA. So if you change like the last base of the 3, the chances of you having a problem with the protein that you're producing is pretty small versus changing the first one in the sequence. And at that point, your chances of having a mistake made in the protein are really, really high. This is what a point mutation looks like. So if you look close here, this is our normal mRNA strand, and the area I want you to focus on is right here where you see this GGC over a glycine amino acid. And if you look closely, you can see as we go down, the first G is the one that's been changed, and we've replaced it with an A, meaning we no longer have a glycine amino acid there. Now we have serine. Okay, now who knows? Maybe that serine versus the glycine has a radical different shape and changes the whole function of the protein. Maybe it doesn't. But at the end of the day, that's about as bad as our point mutations can get built into a piece of mRNA or DNA for that matter. There's one other exception where we can end up with problems at the beginning or end, right? So here we have a stop codon, which basically tells your ribosomes to let go of the proteins that they're making. If you damage your stop codon, or at the other end, the start codon, AUG, now you're in big trouble because there's only one start codon in the entire world, AUG, which creates methylated caps for amino acid sequences that lead to protein. So if you don't have an A, U, G at the beginning of your amino acid sequence here, or your piece of mRNA, that protein will never get made. And if you don't have something like UAA at the tail end of your mRNA sequence, then that protein will never be released and like be able to be used for anything. So that's what point mutations are all about. This is just a quick reminder of the genetic code and the 3 base thing that I was talking about. So if you look closely, there are lots of amino acids in this genetic code table that you can get by using slightly different triplet codes, or I should say codons, because this is from mRNA. So the idea again is that we can alter the mRNA sequence, which is written from our DNA templates, ever so slightly and still end up with the exact same amino acid structure, right? So if you look really closely, there's lots of examples in here. Like here's a leucine, where there's multiple ways to get leucine. Really doesn't matter what we do to the sequence as long as we don't change the C at the beginning. You change that C at the beginning and now all bets are off as to what you're going to get, okay? Frame shift mutations. So the question in a frame shift mutation is what would happen if a single base were actually lost from the DNA strand instead of replaced, but like, this time it's literally going to be kicked out, or maybe we could do the opposite. We could add one at the, beginning or somewhere down the line of our mRNA sequence. Well, the answer to that question is, well, it would literally change the entire reading frame of your mRNA strand and radically change the protein sequence that you're going to produce. Here's why. So if you look at this, okay, we've got a reading frame on this piece of mRNA that's supposed to read AUG, AAG, and then maybe UUU right here, okay, then a GGC, then a GCA, and so on. Well, what's taking place here is one of the UUUs in that codon I just read has been removed, and by removing it, it shifts the reading frame forward, and now instead of reading the codon UUU followed by GGC, well, the reading frame changes to UUG, then GCG. This could give us a complete, total, radical, different protein than what we were expecting or than what we actually need at the time, and can have a dramatic impact on not only the cell but the whole organism. So frame shift mutations, by and large, I'm just going to say are bad. Moving on from there, the mutation would cause nearly every amino acid in the protein after the deletion to be changed, and a mutation in which that single base is added or deleted is again called a frame shift mutation. So while the point mutation and frame shift mutation sound bad, it can actually get worse. I know, as if it, couldn't get any worse. Now we have what are called chromosomal mutations or chromosomal alterations. Now, why are these worse? Well, because these deal with whole chromosomes, which could have hundreds of genes on them, not just one. So through a chromosomal alteration, we could be damaging, disrupting, or changing literally hundreds of genes at a time, hundreds of proteins at a time, instead of just one. That's a serious, serious, serious mutation. So when we think about chromosomal mutations, this is an alteration in a whole chromosome of the 23 pairs or 46 chromosomes that we possess in our cells. So chromosomal mutations can occur in all living organisms, but they're actually much more common in plants than they are in animals. A few chromosomal mutations can be passed on to the next generation, but because the zygote usually dies as a result of the mutation, well, those are kind of few and far between. So going back to the serious nature of the chromosomal mutation, if I damage, half of a large chromosome, I'm going to be missing hundreds of proteins that are necessary for my cell to function normally. And, well, the zygote may not survive that. Okay. So let's take a look at some different types of chromosomal mutations. In the case where a zygote might live and develop, The mature organism could be sterile and incapable of reproduction if the chromosomal mutation has affected the sex cells of the individual that's inherited it. One example of a chromosomal mutation is what's called a deletion. So if you look at this chromosome down here, you can see I've got genes A, B, C, D, E, F, G, and H on this chromosome. And as a result of the deletion, we come over here and you'll notice there is no D gene. Now, I don't know what the D gene was for, but at the end of the day, it's gone. I don't necessarily know what caused it to disappear, whether it was just a mutagen like gamma radiation or a literal mistake in the sorting of the chromosomes and copying them through cell division. It doesn't matter. The point is, the D gene is gone, and whatever the D gene coded for is now not being passed on to future cells, or if this is a sex cell like sperm, egg, or pollen, then it's not going to be passed on to future generations and could result in the death of the zygote. So that's what a deletion creates. This is a duplication. When part of a chromatid breaks off, attaches to its sister chromatid, an insertion occurs, okay? An insertion can lead to a duplication of genes on the same chromosome. So here we have, again, a chromosome with A, B, C, D, E, F, G, and H genes on it. And then as we look at what happens in the duplication or the insertion, I've got A, B, C, B, C, D, E, F, G, and H on the next chromosome. Why is this bad? Well, at the end of the day, you've got more genetic material than what you need, and you could end up producing more proteins of a particular type than what you need. This could cause your cell division cycle to move forward very, very quickly. It could cause any manner of defects. It just depends on what those extra proteins code for or control. So an insertion is an extra piece of genetic material inserted into a chromosome that leads to, all manner of problems based on what those genes control. Next one we want to look at is a situation where you still have a insertion, but in this insertion, we've basically taken a chromosome, we've broken it off, flipped it around backwards, and put it back in. Now, this doesn't sound like too much of a problem because I'm not really adding extra genetic material. The issue here is really only in the order in which proteins might be produced. So as you look at our two examples down here, I've got the normal alphabet load worth of genes, and then here you can see instead of going A, B, C, D, I go A, D, C, B, like that. So where this might become a problem is, again, in cell division. So let's say that A is the gene that codes for the protein that gets me from prophase to metaphase, and B takes me from metaphase to anaphase, right, and so on. Well, if I flip-flop the sequence and I go from A, prophase to metaphase, and all of a sudden I'm met with a D protein, how am I going to get to anaphase, right? Like, it can really disrupt things that are supposed to happen sequentially. I can end up producing proteins at the wrong time and, again, upset the balance of cell division and create all manner of multiple issues for not only the cell but the organism as a whole. Here's another example of an insertion of sorts. This one is called a translocation. So in translocation, I've taken a gene off of one chromosome where it's supposed to be and moved it to a completely different chromosome where it's not supposed to be. Again, this can end up with an individual inheriting extra genetic material that they don't need, or me producing proteins out of sequence. This is a great example of what happens in a genetic disorder called trisomy 21. You might know it as Down syndrome, where a piece of the a chromosome is actually inherited, extra piece of a chromosome is inherited at the number 21 position in a person's genetic code. And as a result, there are all manner of issues associated with that genetic disorder from inheriting just a small piece of an extra chromosome as opposed to a whole one. So these chromosomal disorders at the end of the day can be quite serious. Remember that some mutations just happen, perhaps by mistake, pairing DNA during replication. If that's the case, those mutations are considered spontaneous. Other mutations are caused by factors in the environment. Those things that I mentioned a while ago, mutagens. If you didn't write it down then, now's your chance. So the mutagen is any agent that can change your DNA. It could be radiation, chemicals, even high temperatures. There's a wide variety of different things that can damage your DNA. And remember, when I use the words damaged DNA, I could be talking about a point mutation, a frame shift mutation, or all the way up to whole chromosomal mutations, okay? So these things can wreak havoc on your genetic material, but keep in mind that not all mutations are created equal, and therefore not all mutations are considered bad. Some of them will literally do nothing, whereas others can create everything from cancer to death. So there's a really broad spectrum on what these mutagens will do. At the end of the day, the point is they break your DNA strand, and we have to fix that. So that's where the repair process steps in. Breaking and reforming of your DNA molecule can result in deletions of all kinds. So you might lose a single base, you might lose a whole chromosome. Mutagens like dioxin, asbestos, benzene, formaldehyde, these are commonly found in buildings in the environment, and all of them can cause these types of deletions and or substitution type mutations. So the final thing to keep in mind is that you are well protected in a lot of ways against this type of damage. I don't want to scare you. I mean, your cells are really, really good at finding mistakes and fixing them. The problem is, the more we are exposed to these things, the harder it is for that cellular machinery to keep up. The older you get, the more the machinery sort of wears down and doesn't work as efficiently as we'd probably like it. So you want to try to avoid exposure to these mutagens no matter how old you are, just for the greater good of helping your cells and your body out to avoid the damage to begin with. All right, ladies and gentlemen, I'm Mr. Kennedy, and that's the story of mutation from start to finish. I hope you learned a lot, and I'll see you next time.